Have you ever wondered how scientists grow billions of bacteria inside a laboratory? How do they keep the bacteria pure, prevent contamination, and store them for years? In this video, we'll walk through the complete bacterial culture workflow from preparing media to long-term storage. Let's begin our journey into the bacterial culture essentials. Before we start, let's understand two important terms. A bacterial culture is simply a population of bacteria grown under controlled condition of laboratory, and a pure culture contains only single type of bacterial species. Maintaining a pure culture is important because contamination can completely alter the results of experiment. So pure culture is needed for any biological or any experimental work. So what are the nutritional requirements for the bacteria to grow? Just like we humans need food, bacteria also require nutrients to grow. The most important requirements are carbon and nitrogen sources. And carbon provides energy and building blocks for cellular components, whereas nitrogen is essential for making proteins and amino acids, nucleic acids. Inorganic salts provide important ions and help them maintain their pH, and sometimes carbohydrates is also added into the nutritional media for additional energy. So there are two types of growth media. One, liquid media, which is known as broth, and solid media, which is made with agar. So broth is mainly used when we need large quantities of bacterial cells, and solid media is usually needed when we need single colonies, and we want to have a single colony of bacteria. This is more like a gel like structure, and this is more like a liquid texture. So how the agar is solid media is made? It is made with the help of agar. And what agar is? It is a polysaccharide derived from the red seaweed called rhodophyceae, and it have a power that it is inert and not non-nutrient, meaning it is not metabolized by most bacteria. It simply provides a solid floor for colonies to grow. So that means agar simply acts as a solid support. So this property of agar makes it perfect for culturing microbacteria, as we see here. Now once we have the ingredients and these things for the nutrients for the bacteria to grow, now how we make it actually to grow the bacteria? So what we have is, we have a powdered media of either broth or agar media, and it is dissolved in water, distilled water, which is mixed thoroughly until it dissolves. The dissolved media is then sterilized using autoclave, which is done at 121 degrees Celsius for 20 minutes. So now this process completely sterilizes the media and remove unwanted microbes and ensures that it is the media is contamination free and desired for our bacteria to grow. So now once our media is made, how do we actually plate the cells? So for that we need some aseptic techniques to make the environment sterile from the contamination. So what we do is, we pre-sterilize pipettes and glasswares. That can be done using flame. We light the flame and hit the rod so that the bacteria or any contamination is removed. This is then cooled and used for further streaking or plating. So now how do we culture the bacteria? From the stab we streak the plates. So when we have a new bacterial stab, we using the sterile loop, bacteria is streaked across the agar plate containing antibiotic. Then antibiotic may be ampicillin or kanamycin. The plate is then incubated at 37 degrees Celsius for overnight or around 16 hours. The goal of streaking is to isolate a single colonies that a single population of bacteria is grown from, and each isolated colonies can further be used to culture it in a large scale and for use for the experiments. So now how do we go for liquid culture? After obtaining isolated colonies, we select one healthy colony. This colony is transferred using the sterile loop into the liquid broth media containing antibiotics, and it is also grown at 37 degrees Celsius, 200 rpm, which is revolutions per minute, to provide aeration and ensure uniform growth. This is also done to scale up the bacterial population. So what is the need of subculturing? Bacteria cannot remain indefinitely in the same culture media. Over time, nutrients become depleted and toxic waste products accumulate. As a result, bacterial viability decrease the cell and cause death of the bacterial cells. To maintain healthy culture, the solution is we have to periodically add fresh media to the growth of bacteria, so that it can maintain a healthy population. This process is known as subculturing. Now what if we want to preserve the bacteria for months or even years? The solution is we make glycerol stocks of the bacteria. Typically, bacterial culture is mixed with glycerol to make a final concentration of about 25%. So glycerol helps protect the bacteria from damage during freezing. The stock is then stored at -80 degrees Celsius, preserving both viability and genetic stability. And whenever the glycerol stocks are made, it has to be labeled properly with the strain and plasmid name, date, and which antibiotic it have, and initials means who is preserving the glycerol stock. So let's summarize the entire bacterial culture workflow. We begin with isolation of agar. Bacterial culture on agar plate. Next comes scale up in liquid media. In healthy cultures are maintained through regular subculturing, and finally important strains are preserved as glycerol stock in -80 degrees Celsius. And throughout every step, one golden rules remain the same: maintain pure culture and practice aseptic techniques. And that's the complete bacterial culture workflow, from media preparation to long-term preservation.